Texas A&M University. So I'll be speaking about the Survey of Odontomachus and general observations of nesting preferences in the Commonwealth of Dominica in the West Indies. All right, introduction. So some background about Odontomachus. Odontomachus is Latin for large tooth or large jaw. And these ants are most distinguished by their ability to lock their jaws out at 180 degrees. That's called the trap jaw ant. The jaws themselves are released to snap shut at incredibly high speeds when those hairs are triggered at the base of the mandibles. It's considered one of the, if not the fastest predatory action known to man of any animal. Now, in addition to the jaws being able to snap shut at high speed, they developed interesting escape behavior. So these jaws, with so much force behind them, they can be released at, say, predators or the ground. And by hitting it at such a high speed and force, they can actually launch themselves backwards and away from danger. A prime application, this is like an antlion pit. If you drop Odontomachus in an antlion pit, it will launch itself out of the pit. The antlion doesn't actually do that. And there are some videos online, if you look, you can actually see the jaws hitting the grain sands and impacting at very slow motion, and the ants go flying away. As for habitat, these ants prefer leaf litter, damp soil, rotting logs. They like to stalk their prey, as you can see by the jaws, they're highly predatory ant. And they do not like to be out in the light. So you're not going to find them just walking around and looking. You're actually going to dig deep in leaf litter to find the ant. They can be found in the tropics, subtropics, and as for the United States, they've found to be established in the Gulf states over the past couple of decades. Now, this survey was done during a study abroad, which is Texas A&M's Dominica study abroad. Dominica is an island that can be found in the Lesser Antilles, located there just south of Guadeloupe and north of Martinique. Dominica itself is divided into 10 parishes. The ATREC, which is the Archipelago Tropical Research and Education Center, is located in the St. Paul Parish, which is where the abroad was based in. I did most of my lab work. Now, this is some pictures of the abroad, the ATREC on top left, a creek just below the ATREC, kind of giving you an idea of the environment. It's primary tropical rainforest. The insect vacuum that I wish worked short out on the first day. I wish it had been used. And I have a photo from my camera of the Odontomachus. You can see the jaws open at 180 degrees on the one on the right. All right, here's the methods. As I said before, that wonderful vacuum died on us, so I stuck with saw forceps and manual aspirators. The ants were collected in small alcohol vials and 95% ethanol. Any location the city abroad had the itinerary I traveled to, I attempted to collect. And they're all collected in nest series because, of course, ants, even though the same species, different colonies can be distinct populations, so make sure they are separate even though they're in the same general area. All the information was recorded and GPS was kept on all their locations. These specimens were identified at the species level by dissecting microscope, high-resolution microphotography, and through personal communication between the Andy Suarez lab and the Smithsonian Institute. Now, this map has been reoriented. Please note that north is actually to my right. This is making it a little bigger on the screen. Any location that has a red blip is where Odontomachus was found during collecting. Yellow indicates where they were not found. Total of 14 locations. 10 is where they were found. 4 is where they were not. Now, as a result, the two species that were found on the island were Odontomachus boweri and Odontomachus ruginosus. And as for differences between them, before we move on, at first glance, sometimes it's easy to distinguish them by their colors or slightly change in size. Boweri being a little bit darker, ruginosus having an orange-reddish coloration. But for an authoritative identification, you look at the node on the petiole. If you look on Odontomachus boweri, the leading or the anterior edge is more convex, whereas ruginosus is slightly less convex. And you need that for an authoritative identification. And I just have some of the macro photography I took with Dr. Booley's amazing photography equipment. Here is an boweri worker. Once again, see the 180 degree on the jaws and the trigger hairs. Here's a male, which is a lot different than the rest of the cast. The queen and an A-late queen. As for ruginosus, I only had 2 specimens during the trips. There's only a worker and a queen to look at. Here's ruginosus, it's the difference in coloration, but the identifying character, as you can see on the node, is slightly less convex. And here is a queen, same situation. As for the data, out of all the areas collected, elevation-wise, there was no ants that were found of this genus above 1,686 feet above sea level. And for the 4 places I went to, there were not any Odontomachus found. The trend was, of course, not finding any ants above those elevations. However, one almost at sea level was differing. I'll explain that later in the discussion. As for nesting attributes, as expected, they love leaf litter, damp soil, rotting wood. But out of interest, they were found inside live termite colonies. It wasn't just them raiding, they were actually inhabiting the nest, their brood and everything were inside the mounds. And they were sharing the same space. Bring on to discussion. Now, as I mentioned before, there was one area at sea level that the ants we looked for, they were not found. It's easily explained because the ants, they prefer leaf litter, damp soil, rotting logs, and this coastal area had almost no canopy cover. It's all sandy. It's not going to produce as buildup of leaf litter the ant prefers to live in, so it's expected you're not going to find that there, even though we try to look just in case. What you notice is, from what I understand to be a new record for the island, everyone always assumed it was just a Odontomachus bowery. However, it's also not surprising because the island to the north, Guadeloupe, has a record of Odontomachus ruginotus being present. And being used in the botanical gardens, Brazil being the capital of Dominica, they were importing plants all the time. They could very easily say, hey, we want something from Guadeloupe, and there was a colony of ruginotus in there in a pot. They got pulled over, and they just established on that island. Now, as for the Bowery inhabiting the live termite mounds, I found this interesting because I could not find anything about this in prior literature. People talk about all the time how for many ponderine ants, which is what the subfamily that these belong to, they hunt termites. It's a wonderful food source for them, but never about them actually inhabiting the same space in the colony of the termites. It's always abandoned, or they're just leading raids to acquire food. And I think that's even more interesting because, like a lot of people know, termites have a climate control. The mounds create these conditions that are perfect for the life cycle. And every time I disturb one of these mounds, the Odontomachus were inhabiting the spot just where the ground met the bottom of the mound, the basal part. And if these conditions, as you found the brood, if they're just right for the brood, if they never have to leave the nest and get their food there, and they remain, it's perfect conditions for them. It's like the best deal they could ever have. They don't have to go anywhere. Everything they need is right there, except for when they have alates and go out and spread. So this would be something really great for someone to actually investigate and study specifically in a further study. And lastly, the reason for the 95% ethanol is because there is a postdoctorate at the Smithsonian Institute. He worked with Andy Suarez in the social insect lab, and he's doing a biogeography for the Odontomachus across the entire Caribbean. And he did not have any specimens from Dominica, and I kind of filled the gap for him. I'm very glad to contribute towards that. Of course, you need 95% ethanol to be good for DNA sequencing. Brings us down to acknowledgments. I'd like to acknowledge Dr. James Wood and Thomas Locker. They led the study abroad program, and James Wood provided a lot of the camera equipment to make those pictures possible. Andy Suarez in the Social Insect Laboratory, Fred Larrabee, the postdoc at the Smithsonian Institute, Ryan Selking, Shelby Kilpatch, Robert Callahan helped collect a lot of the ants. The ATREC staff, who took care of us during our stay for the study abroad. Stefan Durand, Dominica's Forestry Wildlife Parks Division, they gave us the permits that allowed us to export the specimens. And of course, most importantly, Matty Cartwright, who is the organizer from the university that makes the trip happen, and Texas A&M itself and the Dominica Study Abroad program. And with that, I now come to questions.
It looked like some of the females had a pretty good stinger. Can they sting as well?
Oh yes, you see, that's why I wanted the insect vacuum. I was actually afraid of that beforehand, but I observed, I didn't run in there. You have to really be messing up while collecting to get bit or stung by these because they'd rather avoid you. You can stick your hand down in there, unless you grab one, you're not bit or stung once, but if they ever do, I'm told it's pretty nasty. I wanted to mention it earlier, they call them the tac-tac, the locals, because of the jaw snapping together. People avoid them. People thought I was crazy. I said, can I find some tac-tac? They looked at me like, why on earth do you want these ants? Any other questions?